No?
Yeah, you go. So Mendel did, Ashley, y'all with me? Mendel did what he called a test cross. Let me explain to you what a test cross is about. That's the next section of reading that you had. And suppose we had a tall plant. If you looked at that plant, could you tell what its genotype is?
Yes.
You can tell just by looking what the genotype is?
No.
No.
What's the possibilities for its genotype? 10 points. If you can write down the two possibilities of the genotype of the tall plant. 10 points if you can write that down for me. What are the two possibilities of the genotype of the tall plant? Blue correct. Peach correct. Orange correct.
Comment. It's not white.
White correct. Purple correct.
What a throw.
Green correct.
Hello. That was great.
Yeah.
Y'all got it. It's big T
That was so mean.
or big T little t. So you can't just tell by looking at it. It's one or the other. So Mendel invented what he called the test cross. So you can figure out what this parent is. What a test cross is a cross with a homozygous recessive plant. A cross with a short plant in this case. That's little t, little t.
There's no such thing as heterozygous recessive, is there?
There's not. If Mendel crossed this unknown plant, we'll just put a question mark, we don't know which one it is, with a little t plant, a homozygous recessive, you can imagine there would be two possibilities for the kids. If all the kids were tall, then what does that mean this parent was? Was it this or was it this? If every kid were tall.
Left.
It's big T, big T. But, on the other hand, if half the kids were short, you know what that means the parent was?
Big T, little T.
It wasn't big T if half the kids were short. It must have been big T, little t if half the kids were short. You see what I'm saying? He could tell by crossing it with a homozygous recessive and then looking at the kids, he could tell which one of these two it was. He would say, oh, it was this one, because it had little short kids. It couldn't have little short kids if it were this one. If they all ended up tall, then it was that one. He could tell which one it was. He called that a test cross. Isn't that cool?
Isn't that a little excessive just to try to figure out what the genotype is?
Yeah, but he didn't have anything else to do. Just hanging out in the monastery.
He was a monk?
Yeah?
Where was he from?
Austria.
Pretty well.
You know where that is?
Yeah.
Small Australia.
Not Australia, Austria.
I know.
Like, questions about any of this? Test cross? Understand test cross? Pretty simple.
Hold it. You don't need it. I got it.
Now, the last part of the reading is an honors section. And it talks about how Mendel wanted to consider not only one trait, like tall and short, he would consider two traits. Like not only considering tall and short, but also considering round and wrinkled. And it made Punnett squares a little bit more difficult. Here's a Punnett square of what Mendel called a two-trait test cross, where he's not only considering round and wrinkled seeds, but he's considering yellow and green seeds. And there's a lot of different combinations. You can have round yellow seeds. You can have round green seeds. You can have yellow wrinkled seeds. You can have green wrinkled seeds. There's different combinations here if you're considering two genes. Yes?
Why do they write it little r then big R? Bottom left, or like the left, yeah.
I don't know. I just noticed that when you
Wouldn't
said
it?
it right there.
I mean, it's the same thing, but
You're
wouldn't it?
supposed to write the big one
Yeah,
first, so they
that's what I
screwed
thought.
that up a little bit there. I don't know where this picture came from. Good point. So what I want you to do is I want you to understand how they did a big Punnett square like this, how that comes about. And to understand it, you really need to understand the genes. Let's consider two sets of genes. Tall, a gene that's for height, tall or short, and a gene for shape, round or wrinkled. Let's consider a plant. Let's consider a plant that's big T, what's that mean? Is it tall or short? Tall. And big R. What's that mean that seeds are?
Round.
Round seeds. You'll see the round peas before, right? And what if we mated this plant that's tall with round seeds? What if we mated it with a short plant with wrinkled seeds? Have you ever seen the little wrinkled peas? Those are good. We made a tall round pea plant, tall, and I'll draw the pod here, with round peas, mated with a short plant, and I'll draw a pea pod for that, with wrinkled peas. It's fun to draw wrinkled peas. Wrinkly peas. A tall round with a short wrinkled. You'll see what we're doing here?
Yes, sir.
What do you think the next generation is going to be?
10 points.
Tall is dominant, round is dominant. 10 points if you can guess what the next generation is going to be.
Could be an oracle. No. Key or future.
What's my future?
10 points if you can guess what the next generation will be.
How many plants?
100 plants.
Star Trek.
What did you say?
Let's try it out.
Just a guess, as you shouldn't have to do any work on this. Phenotype.
Yeah.
Oh, wait.
What do you think the next generation will be? Yes! Peach got it.
Come on, white. Go.
Alright.
We can beat you tonight.
Orange got it. Phenotype.
Yeah, well.
Blue got it.
Oh, okay.
Green got it. Purple got it. White, anything?
Yeah.
White got it. Good job. They're going to be tall and round. Tall and round. That's in their genotype will be big T, big R, little r. Can you see why their genotype would be this?
Yes.
They get a tall from the dad and a short from the mom, big T, little t. They get a round from the dad and a wrinkled from the mom, big R, little r. But tall and round dominate over short and wrinkled, so you end up with tall, round plants. But what if you mated two of these together? How do you figure out what the kids are going to be like?
Punnett squares.
You have to do a big Punnett square that looks like that. There's a whole lot of different possibilities. And those possibilities come from meiosis. So let's jump back over here to meiosis. Do y'all remember these chromosomes, these sets of chromosomes? Remember there's one set from the dad and one set from the mom?
Oh yeah.
Pay attention here, this isn't easy. Let's suppose that the dad gave a big T gene. And the reason why I put big T on this other one was because, do y'all remember that this and this are exact copies of one another that formed an S phase? Hannah, you with me? This and this are exact copies of one another. So if this had a big T gene, then guess what the white one has? If it's this heterozygous here, it's not going to be a big T. You know what it's going to be? A little t. That's why this plant is big T, little t. It's got big T on one chromosome and little t on another chromosome. That's why it's a big T, little t plant. Does this make sense? What are they calling me about? Willis? Hey. Okay, will do. In fact, Thomas Grimm, you have to return. Is Thomas Grimm in here? You have to return that, not yet. At the end of class, you got to return that to the library. All right. It's big R, little r. Guess what? That's also located on the chromosomes. Big R on one chromosome, little r on another chromosome. Can y'all tell that the organism that this cell belongs to is big T, big R, little r?
Yes, sir.
That's this. So the organism that this cell belongs to is tall with round P's. Y'all see what I'm saying? And when we go through meiosis and we make sperm cells, what we're going to do is line these things up in the middle. Line them up in the middle. Yeah, they switch pieces during crossing over. And then separate them out into sperms and eggs. Remember this process? And the sperms on this side are going to get a big T and a little r. And the sperms over here are going to get a little t and a big R. So that's two possible sperm cells that this organism can make. And I'm going to draw them in little circles here with little tails, because sperms have tails. They look like balloons, don't they? But you can get a big T in a sperm and a little r in a sperm. Let me draw it like this. You get a big T in a sperm and a little r in a sperm, like this sperm that forms over here. Or you could get a big R in a sperm and a little t and a big R in a sperm. So you could get a sperm like that. There's two other possible sperms. Do y'all remember when we line these things up in meiosis? Do y'all remember they could switch like that? Remember what we called that? Independent assortment? And then go to opposite sides? And then you would end up with a big T, big R sperm, a big T, big R sperm, and a little t, little r sperm. There's 4 different types of sperms that you can make from this plant during meiosis. 4 different types of sperms, and that's why we have this big Punnett square, because you can make all these different types of sperm cells. There are 4 different types of egg cells that the female could make. She can make a big T, big R egg, a big T, little r egg, a little t, big R egg, or a little t, little r egg. Look at all the different possibilities for sperms and all the different possibilities for eggs. This is why you get these big Punnett squares. And how do you put the genes together in these big Punnett squares? You put the two T's together to see if it's tall or short. What's that one going to be?
Tall.
Tall. You put the two R's together to see if it's round or wrinkled. What's this one going to be?
Round.
That's a tall round. There are 16 different possibilities for the kids. But that one's tall round. What is this one? Big T, big R, big R. What's that?
Tall round.
Tall round. Big T, little t, big R. Someone besides Joe.
Tall round.
Tall round. Big T, little t, big R. Someone who hasn't answered yet. Tall round. Big T, big R. Someone new.
All around.
Tall round, yeah. If you hadn't answered, take your turn here. Big T, little r. What is that? Tall wrinkled. Big T, big R. What's that?
Tall round.
Tall round. Big T, little r. What's that?
Tall wrinkled.
Tall wrinkled. Look at all these possibilities. Big T, big R. Do y'all see how I'm doing this? Big T, big R. That's what goes in this box. Tall round. Big T, big R. What's that? Tall round. Little t, big R. What's that? Short round. Eh? Little t, big R. What's that? Short round. Big T, big R. What's that? Tall round. Big T, little R. Someone else. Tall wrinkle. Who hasn't said yet? Little t, big R. Someone who hasn't said anything yet. Short round. And final one. Little t, little R. Short wrinkled. Yes.
Use the restroom.
Yeah, well, wait, we're not finished yet.
Okay.
Let's count up the phenotypes. There are 4 different possible phenotypes. Let's count them up. Let's count up what the ratio would be. Let's start with tall round. Let's count how many of the 16 are tall round. Tall round. Tall round. 3.
9 of them.
4, 5, 6, 7, 8, 9. Y'all see that all those are tall round? 9 tall round. How many short round?
1, 2, 3.
Short round, short round, short round. 3 short round. How many tall wrinkled?
3.
Tall wrinkled, tall wrinkled, tall wrinkled. 3 tall wrinkled. How many short wrinkled?
1.
1 short wrinkled. That was a lot. Mendel called this a 9 to 3 to 3 to 1 ratio. He called this type of cross where you take an organism that is a heterozygous for 2 different traits and you cross it with itself. He called it a dihybrid cross. And he predicted that if he did a dihybrid cross, he would get a 9 to 3 to 3 to 1 ratio of kids. And that's what he got. He grew if you grew 1,600 plants, you would have 900 tall round, 300 short round, 300 tall wrinkled, 100 short wrinkled. And that's what he got. Isn't that cool?
Bro.
That's called a dihybrid cross. Now that last section about dihybrid crosses, I put that for the honors kids because I wanted you to understand that. I'm not going to give you a problem where you have to do this. I just wanted you to show you how he did it and how he worked out with all these different possibilities. Yeah.
How would you get all the genotypes from just the equation?
All the genotypes.
The left ones, like on the outside.
These?
Yeah,
From this?
yeah.
Okay, so you just take one of each pair goes into a sperm cell. So you can take one of those two, pick which one you want.
Big T.
Big T into a sperm cell, and one of those two.
Big R.
Big R. So that's one kind of sperm cell.
Mm-hmm.
Are there any other kinds?
Mm-hmm.
Yeah, you could take a big T and a little r. You could take a little t and a big R.
Okay.
You could take a little t and a little r. Those are all four possibilities.
Got it.
See what I'm saying?
Maybe the rest.
So you take all the possibilities for one parent and all the possibilities for the other parent, and that's your square. Now you can go to the rest. What y'all think? Okay, for 10 points, let me ask you this. If I have a P that's this, tell me what its phenotype would be. 10 points. If I have a P that's that, tell me what its phenotype would be.
Wait, for what? The TTR? Yeah.
What's the phenotype? Yes, orange is correct. Blue is correct.
Toby, you better.
Green is correct.
What?
You better.
Peach is correct.
No, I have no idea.
White is correct. Anyone else? No? I figured everybody would get this. That's tall. That's wrinkled. Wrinkled. Tall. It's a tall, wrinkled plant.
Oh, that's all you have to write?
Yep.
Yeah.
Does that make sense? All right, here's what I want you to do. You got 10 minutes left. I want you to read this chapter. If you haven't read it yet, read it. If you've already read it, read it again. And there's going to be a quiz tomorrow on it. So make sure that you have read it and understand it and have notes. If you can use them on the quiz tomorrow, you might as well do that.